next Tuesday. Today we will continue, or we will begin on the secondary structure prediction. Or we continue, so we have already done that. Next Thursday is Corpus Christi, so no lecture. Another announcement. We are looking for IDP students. This is a project named Carry Your Genes. It's a collaboration with a company. It's about personalized medicine. It's about the ability to, in fact, put your genome onto a stick and carry that around to the doctor and other places. It's an Android smartphone app, and we want to simply explore how is that feasible? How much amount of data can we put on it? What are the technical challenges? So this is a proof of principle, if you want. The contact is Lothar Richter, Daniel Starr, and Mark Hoffman here. Those are the two websites. It's a paid TV job for 1 month in combination with an IDP project. So if you want to know more about it, come to me, talk to those guys. Okay. Recap. And actually those slides are not in the set on the web. Should I put them up? No, they are not on the web because I just put them in just a few minutes ago. In fact, that's the reason why I'm late. I will put them up. Recap on secondary structure. But if that is not for you, we are looking for IDP projects. Anyway, again, sequence determines structure, determines function. In principle, we could predict structure from the sequence alone, but it's a challenging problem. When we compare sequences, when they're very similar, we can easily do by pairwise comparisons. When they get more dissimilar, the problem gets harder, and we need to compare sequence against profiles, so against families. And when it gets even harder, we need to compare profiles against profiles, which essentially means families against families. The good news is that by matching, so I never got back to your point. If I have my sequence and I find in uniprotein protein that has known structure, then I know that the structure for my protein is known and I can do much more. If I don't, I can apply comparative modeling. Comparative modeling essentially uses the idea that two proteins that are very similar in sequence have a similar structure. If for one of them, then my structure is known, not for my protein, but for one that is relatively similar, I can infer through comparative modeling the structure for my protein. I can apply that today for over 25 million proteins. Bad news is, for most residues, most proteins, we still cannot predict structure with comparative modeling. Yes, we can do it for many, but for most of the pie continues to be empty. That means that in this language of 3D, 2D, 1D, essentially 3D is no longer accessible to us. Okay? So today we begin with a series of lectures that go into what if we completely get humble and forget about trying to predict 3D structure. Could we at least predict secondary structure? What can we do? And again, so we started that last week ago. I talked about the first and second generation methods. I said the first generation method essentially compiles a probability for a particular residue to be in one of three secondary structure states, helix, strand, or other. And the second generation methods, they essentially compile a probability that depends. What's the probability given that here you have a leucine and all of these 6 residues on either side, or 3 on either side? What's then the probability for the leucine to be in a helix? That's the second generation method. And I told you that those about the time frames here, but the problems are they're not 100%. Accuracy, maybe there is a max because secondary structure is not completely local, not completely determined locally. In fact, mapping to this explanation is that beta strands are predicted almost at levels of random, and beta strands are less local. As you see, the formation of the hydrogen bond here is between things that are further apart in sequence, while in the alpha helix they are I and I plus 4, so close in sequence. I and I plus 4 means that if I have 5 residues in my input to a conditional probability, there is a hydrogen bond between these two if that is a helix. For the beta strand, the hydrogen bond may be with something very far apart, or in this particular case here, it's even further away. And the other problem is that there are short segments. I briefly introduced the machine learning device of neural networks. An input the layered networks here as a device that is supervised learning. So you present examples, and from these examples it tries to map the data that you give it, input, output, connection. The three parameters are the connections. You change the connection such that the output that you, in fact, that's supervision. You know what you want here. You know what you have. So you adjust this connection such that it is an optimal match between what you want and what the system gives you. You begin with a random initialization of these connections, goes somehow in an error landscape downhill. An expansion of this is introducing hidden layers. The rest of the story is the same. And then there's this issue of training time, performance here, green and blue, and overtraining, and the testing. What are these two curves here? Well, overtraining and cross-training. So it's important here that this is not the test set, or that this is not the set for which you essentially write your paper. Typically in papers, when you apply machine learning, you almost never present training data. Now, this curve that I show here is again something that you typically don't present in a paper, because what you typically present in a paper is the data set for which you will assess how well the method works. And that is not this one, because this one here will be used to determine when you stop. It's a bad idea to continue training beyond that point, because the blue goes down. The generalization set, if you want to call it like that. So you somehow do early stopping, you stop here, and that means the blue one is actually a cross-training set. Again, it has different names in the community, but the important thing is that is not the set for which you report performance. You need a three-way split in the cross-validation. In real life, the performance does in fact sort of see maxima here, but you see it's way less extreme, way less pronounced. So this is what previously was blue. And on the other one, this is the training, and this is the cross-training set. Let's get back to the secondary structure prediction. So we have this fancy neural network idea, and we have a problem of secondary structure prediction, that we cannot predict 100%, that we don't get beta strands right, and that segments are too short. How will we essentially use this neural network to predict secondary structure? Well, the architecture is very simple. You use input units, output units. The output units are helix, strand, other, the three states of secondary structure. And the input here is defined by, in this particular example that I show, 13 consecutive units. So 13 consecutive amino acids. Say this is amino acid 1, 2, 3, 2 through 13, right? And the network is supervised, means that you train the system to predict the secondary structure of the central residue, which happens to be a proline, a P here, which happens to be in the state E, extended or beta strand. Okay? These data for training you get from DSSP, from the method that assigns secondary structure from 3D coordinates by tracing hydrogen bonds. I talked about that. Now, what this one hides here is not 13 input units. What this one hides is, in fact, here's a blowout. For every one of these blobs here, I have, in fact, 21 units. I have 20 amino acids. What is the 21st for? So this is, in fact, 13 times 21 input units. What I believe the 21st one is for. So again, I'm going to have this window. I'm going to slide the window through my protein sequence. I begin with the first, second, and these are different examples, right? So for a protein of 100 residues, I would have 100 different examples, right? But now, why do I need a spacer? And again, this thing is supposed to predict the secondary structure of the central residue. Do you begin to see the problem? Can you be a little louder?
We need a window of odd numbers because the value point even will not be.
Even and odd, that's a great guess, but I'm not entirely sure what you're after.
Which in the sense, if it is 21, you know what is the middle of that.
You are a level beyond. Yes, I believe what you are addressing here is the epsilon of the problem, but not the major part of the problem. That could be true what you say. And this, what I like in your thinking is you think about symmetry, which is what we do usually, and very often in science it helps you to think about symmetry. That could be a problem. Actually, I tried this out many years ago. Turns out it's not a problem. But there's something much, much more dramatic here. That is wrong if we had only 20 input units. Again, the point is, your prediction for proline is the central of the window that you slide through. You don't see it? Exactly. So, no, well, you're funny. You go in that direction. But let's begin with 1. With this system, I cannot handle residue 1. I said that the D here is the first residue. If the D is the first residue, and P, in fact, in this particular case, is the 7th, how do I handle the D? How do I ever make the D slide into the middle of the window? Right? And the answer to that question is, that is where I use my 21st unit. This is a spacer unit. So when the D sits in the middle of the window, this 6 amino acids before it, I'll simply set to this 21st unit. And the same when you slide to the other end. That was your end of the problem, right? Okay. So, is this going to improve? Is this going to be better than all the methods that people had been developing until 92 when this was first done? What do you believe? It's a fancy device. So far people had, to that point, so again this is 23 years ago now, but then people had only used simple statistics. Conditional probabilities is not that simple. The formula to work out those formulas are very non-trivial. But this is a different device. Do you believe it will improve? I can tell you, so I worked very hard on this. I actually worked years on it. And I was absolutely confident that would improve because of the modern technology. Then, okay. And the answer was, it gave 62%, which roughly was exactly what everybody else had. So by just plugging in a new device, in fact tailoring it even to the problem, that alone is not enough. And I tried all kinds of things that had pertained to the symmetry, pertained to how to use the input, and I didn't get much beyond that point. So just doing machine learning is not enough. Then I observed something. Namely this. What I show here is the training time, performance. And I show that for 3 different states. Other in green, red is helix, strand is blue. Does anybody see anything? Can you describe can anybody please step forward and describe what you see? Three curves. So I see 3 curves. More than that, tell me more.
The blue one is lower, but it's the only one that improves in a certain way.
So there are a couple of things. Here's another thing you have to learn. When you look at data, describe it to yourself first. And begin, it is trivial and you may consider yourself dumb. But this is often how you discover things that... so first you describe simply that there are 3 curves and they're behaving slightly different. Blue behaves differently. And somehow red and green alternate in some sense. All of these things you may see, but just sit down and say them to yourself or write them down. Maybe that triggers an idea. And whenever you look at new data, try to do that. So again, there is some alternation between red and green, and only blue somehow rises because red and green are pretty much high in wherever they get to. So between 1 and 10 one order of magnitude more of training, and there is not a big change for red and green. But a big change happens only in blue. So The error function here that I show is simply that I try to optimize the number of residues predicted correctly in either of those three states. Does that give you any... How would you react when you see this? Okay, blue is beta strand. The training for blue takes much longer. Any idea why that could be the case? So there's an obvious explanation that you can give that I will attack you on. And there's one that is right. That's the one I attack. So if that would be the explanation, that is what everybody believed in the field until then. And actually today still, there are most textbooks still claim that. And there's some truth to it. But if that were completely the explanation, you would never learn them. That's testing data here. It's not the training data. Does that give you a hint where the real story lies? Let's look at it differently. When I look at the database and ask what fraction of all my residues is helix, strand, other, then that's what you observe. When you ask what of my correctly predicted residues is helix, strand, or other, then that's what you observe. And for beta strand, that is exactly what I talked about, close to random. But do you see something that jumps at your eye? These two things, these two pies look very similar. What you predict, the fraction of strands that you predict correctly, is very similar to the fraction of strands that you see in the database. And that, in fact, would explain why it takes so long. Because there are so few there. The samples in the neural network are presented randomly, one at a time. That means if about 1/3 of the samples are beta strand, then only about 1/3 or less than 20% are beta strands. Then every 5th time a beta strand residue comes. So it has to do a lot before it gets enough samples. How could you change that? Think about a hack. So what you want is something like that. How could you hack that? If that explanation were true, if it had just to do with the occurrence, the number of the frequency. Any idea how you could hack to make that happen? Well, typically what I do, at every single step, I randomly choose one of my samples. Now what I can replace this by is, it's called a balanced training, at which I in fact have one example from each of the 3 classes. So rather than sampling helix, strand, and other according to the frequency in the database, I'm now going to force the sampling that presents them equally often. At every single time step, I will present one helix, one strand, one other, which implies that I completely oversample strand, because it's less often in the database, right? I sample it more often than it actually occurs, which I call balanced because I want a balanced prediction. If we do that, this is unbalanced, and this is balanced. Okay? Yeah?
Can the number of training data be increased to increase the performance?
Yes. So now we get into the reality of biology. So the question for those of you in the back who may not have heard it, the question was, can we increase the number of data points? And would that in fact increase the performance? When you apply machine learning, this is the first question you have to ask yourself. The way to do that, to address that question, is you will have a data set. Say that data set is called 100%. You take, say, 50% of the data out, and you try your machine learning on the 50%. maybe take 10% of the data away as a sort of test set. Okay, and then you increase 50%, 60%, 70%, 80, and then 90, and you see how the performance increases. If the performance doesn't increase, that gives you a clear idea that in fact you have enough data. Most of the time, at least in this field, in this particular case, in this particular experiment, what you will see is that you need more data. And most of the time you will see that there clearly is an improvement from 80 to 90, and that you, most likely if you had 120 or 200%, twice as much data, you would do better, but you don't have it. So typically in biology you use all the data that you have. Alright, in this particular case now, what we get is a method that at the end of the day predicts these three classes completely at an equal probability. The overall accuracy now is 60%, not 62, because I force the system to do strand better, but since strand actually is less often observed, on average I do worse. Now this is remarkable, because you in fact see an example that you can force, if you understand the language of that machine learning device, you can force the machine learning device, in this particular case you just look at one aspect of the training phase or the development phase, and you can see that you can solve a problem that was there before, and in fact you can completely turn around the view that biology has. The assumption was beta strand cannot be predicted very well because it's less local. It is true it's less local, but that is not the explanation, because I can force a system to predict them all equally well. And in fact, this 60% that I showed, the method on that 60% was better than the existing methods that didn't use neural networks at the time of the development. So not always when explanations sound very reasonable and come from experts, they are right. In this particular case, the machine learning device allowed to unravel this. Now let's get to turn to the next problem here. The next problem, so one problem we solved here. We did not solve the 100% problem. We solved the problem with the 40% for beta strands. We're doing better on beta strands. But now we have another problem. We have a problem with short segments. Short segments, again, one argument was that they are sort of cut apart because they are, oh, wait a minute, I got these wrong. The short segments, the example is here, my talk is messed up. Typical example of a prediction of a beta strand is this. A helix about 4 residues, a beta strand of 3 residues. Observe the 6 and 10, 11. So here's a typical prediction method, and remember I said, again, this up here means observed secondary structure. Blue is beta strand. That's the typical predicted secondary structure. H is helix, red. Blue is beta strand. Spaces means other state. And you see that you have, in fact, 1, maybe 2, 3, 4, 5 strands, and here I made a mistake in labeling that's a helix and a strand. And in the prediction I have many, many, many more units, and it's unclear what that means. It's unclear how to use that prediction. And that unclarity because of short segments leads to that prediction. Now where does that come from? And again, the neural network does the same thing. Every statistics method, every method that existed. So when I started working on this in the 90s, at some point I had counted 200 different methods that had predicted secondary structure. And all of them essentially had the same problems. They used very, very different methods, but none of them really addressed the nature of the issue, such as with the beta strand story. Here's another story. Where does that come from? In my universe of machine learning on neural networks is very clear. What I do to train these neural networks, so the machine learning devices, is I go down in some error landscape. In order to go down in this error landscape, I present one example at a time. Presenting one example at a time, I actually have to pick them randomly. If I did not pick them randomly, if I essentially went down the sequence, 1, 2, 3, 4, 5, then I would include or introduce local correlations that I really don't want to have. That is, let's call it the symmetry argument in some sense. You put bias into your training that you always want to avoid. So you clearly have to go for some random chop of all the data. Now, the moment you go for a random chop of the data, the problem is that moving the window by 1, you see the correlation of hydrogen bonds. But if you chop, if at one time point I present an example from one protein and another next time point from a very different protein, this thing never sees correlations. right? Yes, there are correlations. When I have the middle residue number 15 and 16 and 17, they may be correlated. But this thing never sees that because at different time, at the next consecutive time points, continuous, it sees samples from very different places without that correlation. How could I put that back in? Do you have any idea? Again, I cannot train in a way where I do 1, 2, 3, 4, because then I introduce bias, and actually it really goes wrong. I tried that. So I need a better trick to somehow introduce correlations. Any idea how we could do that? There are many solutions. That's a good idea. So let's call it, that's the hack idea. You go from symmetry to hack. I'm smiling because symmetry is a principle and this really is a hack. So let's repeat the idea. The idea is, I said I cannot do 1, 2, 3, 4, 5. So the hack idea, well, if I cannot do 1, 2, 3, 4, 5, can I at least do 1, 2, 3? So can I look for local stretches? And that would help some. That would work in some, but it's actually not good enough. The effect is too small. Better idea. So again, the problem is that I want to bring into my system sees, let's say, 5 consecutive residues. It predicts the secondary structure for the middle finger, and it does not see the correlation between the prediction of that middle finger and moving on when this one here will be the central residue. How can I bring that correlation in? That is a hack. I tried it actually, it's not good enough. It had a measurable effect though. Any idea? So I predict the secondary structure for the central residue. No idea? There are many ways in which you can do that. Here's one. In this way, you essentially have a cascade of, in this particular case here, neural networks or machine learning devices. And you can do whatever you want. But the important thing is that now I have this neural network that I showed you before, where the V is at the center. And I use the output that I predicted before, the 3 states, as an input to another layer, or another neural network. Okay? And then what I show here is essentially 3 consecutive residues. For this neural network, V is the center, P and A, and now P of the second level will be the center. Why do I use 4 units, although my network predicted 3 states? Sorry? The same as before, as the 21st unit, we need a spacer. Again, we're going to slide this through, and in this second layer network, I'm going to train this as the central residue. If that is the central residue, the V, say, that would be the first in the sequence could never become the central residue, so I need a spacer. Same as before. So the second level here, in this particular case, has just, in this, what I draw here, 3 times 4 input units. Output is exactly the same as before. Data sets are the same. When I do that, this is what I get. This looks much more similar to what I observe. When I look at the length distribution, I see there is some overrepresentation of short things that I miss, but overall, that's the length of a segment predicted here in blue and red observed. And you see that these distributions here are not that different. of all, the distributions look fairly similar. So you get essentially the length right. By the way, the first method here that used what I called structure-to-structure network was, in some sense, invented by Sejnowski. And Sejnowski was the first publication that applied the neural network to protein secondary structure prediction, Chan and Sejnowski. And the interesting thing is that Sejnowski used the second level to improve the performance, and that didn't work. They had overlooked that it works for something totally different. Which, in fact, because they had overlooked that, is why many years nobody else tried this. The real breakthrough was to discover that there is something in that solution that does work. Okay, you could also have more output units. So rather than having 3 output units, you could have 9 and introduce correlations like that. Or you could put the output back into the neural network. That's called recurring neural network, and Gianluca Polastri and Pierre Baldi have worked an example for that out. So in this particular example now, so I flipped what I showed before upside down. So now the input is here, written here, and the output of the 3 states is up here. And now essentially I feed back the output here into the input. It's the same idea. This, however, is more generic. This is much cleaner than the hack of the 2 cascaded networks. It's a generic solution to this problem. It has the downside, is a little bit more difficult to train. But this is clearly the better solution. And it goes back to Paolo Frasconi, by the way. Now, I solved many problems. I solved the problem with beta strands, I solved the problems with short segments. But essentially, I still have not improved overall performance. I have made, with these neural networks, I made the secondary structure prediction methods much more useful. And by all means, that was a breakthrough. But we still did not improve the overall performance. The overall performance, somehow, the problem is, I don't have enough examples. Or, put it differently, I can't put enough information into my system. How can I put more information in? Do you have any idea? Oops. You saw that? You're fast enough? Well, even if you saw that, what does it mean? So some here in the room have an advantage because I made a mistake with my animation here. Why is that actually? Because I clicked twice. How can I put more information into the system? Okay, let's discuss first the obvious solution. The obvious solution is making the window bigger. My problem is not well defined. Somehow that may have to do with the fact, after all, that the formation is only local. Making the window longer. It doesn't work. Okay? It's not that this is obviously wrong. We all tried that. It didn't work. Why not? Why do you believe this doesn't work? Oh, sorry. I give you the explanation. I give you the observation that increasing the window from, for instance, 13 to 21 does not help. How would you explain that observation? If you're a student, or if you found that out and you had to present it to your supervisor, what would you suspect as a reason? Why is that the case? And you don't actually, again, the explanation that I'm looking for here has nothing to do with secondary structure prediction or with computational biology. It's a very generic suspicion or a very generic statement. What's obviously wrong? If you, on the one hand, you know that you don't have enough information in your input, and on the other hand, you observe that making the window longer is not helping you. What does that mean? It means that what you put into your system, in addition, say from 13 to 21, the additional 8 residues there, what you put into the system on top But this did not help you for the problem. And that is because, yes, distant information is important, but most of the distant information are not important. In fact, they introduce bias. How come? And that we have to, I'm afraid we have to go back to the beginning of my talk to explain one important aspect of why that is the case here. Here it is. Ultimately, the reason is this is a contact map or a distance map. So the 2D presentation of protein structure. Sequence runs in that direction, sequence runs in that direction. Dark means two residues are in contact. You see, there is structure in this contact map here. Close to diagonal is local in sequence, off diagonal, so these are non-local contacts here, symmetric. Non-local contacts, these are the important ones. These are the ones that are beyond what you could put in local windows. But you see that as far as you move away from the diagonal, it's relatively thin. Most of the points that you see, you don't see. So most of it is white. Meaning that most of the long-range contacts are not forming. There are a few that form. They are essential for the way the structure looks. But most I and J that are distant, for I and J being more than 10 residues apart, are white. And that actually is exactly the bias that you get when you make the window longer. You bring too many things here that bring nothing. They bring only bias and they completely confuse your network. And if you're lucky, you don't do worse, but actually if you go very long, you do much worse. Because the bias is actually beginning to dominate. And that, again, has nothing to do, my explanation why that is the case, has to do with secondary structure prediction, or with protein structures. When you observe that you need more information in the system and that yet making the window longer doesn't help, then the explanation that there is some that there's too much bias added is generic and will work in every context. Okay, we are still at this point. How can we put more data into it? And the answer is evolution has the data that we want. What do I mean by that? At some point I told you about the HSSP curve. Number of residues aligned, percentage sequence identity. If two proteins have 100 are aligned over 100 proteins. So again, the question that I asked at the beginning today, when I begin with a sequence, I search my database, UniProt for instance, and I run an alignment method and say I find 100 residues that are identical between my protein and say one that is in the PDB, for which I know the structure. And over those 100, at least 33 are identical, have the same letter that is, then I know that these two proteins are related and they have similar structure. Sorry. Blue means I know they have similar structure. Okay? Now, fact. And again, let's repeat an important part of this curve here. Having a rough idea how this curve looks is also an important point of some examines. One important aspect here is that if you have two short alignments, you cannot tell anything about structure, because 11 mers can be either way. This is the point where it reaches 100%. And then there's some saturation here. But let's get back to the 100. 33 identical, and I know they have the same 3D structure, essentially the same 3D structure, or similar 3D structure. That means I can change 67 residues in 100. Now, if I went into a lab and I changed 5, most likely what would happen is that I would not create a similar structure, but that I would create a protein that will not fold. How is that compatible with what I show here? I say that I cannot change, or actually it's not that easy for me to change 5 and 100. Many of my solutions will be wrong, or a definition of wrong is I will not get a protein that folds or that does not have the same similar 3D structure. While here I have an observation, everything I observe in nature has a similar structure. How can these two things be true at the same time? the answer to that question has a lot to do with pondering about what evolution means. It's ultimately as simple as all the ones that will not fold, that will not work, we will not observe in nature. All the things that I could pick that go wrong, they will not be seen in this curve. They die. This is the principle of evolution. What you see is a tiny subset of the possible space. And that tiny subset is biased, but it works. That's the bias. And the remarkable thing about life is that many aspects of it are so unlikely to happen. The neutral change is very unlikely. That you change 5 residues in 100 and don't mess up is very unlikely. Yet everything we see can change 67 because we had enough time to try out what survives. And this system, in exploring all the possibilities because time was long enough, goes to finding very unlikely solutions. Now it means something else now. It means that the particular position at which you can change contains information about the structure. You cannot change at every position at equal probability. That depends on what you have. That depends on how your protein looks. Example here. This is just one important protein, the SH3 domain. In the SH3 domain, this is an old alignment from over 20 years ago where you already had such a large alignment. The important point here is there are 4 lightly labeled columns where all the sequences, all the representatives of this family essentially have the same residue. They're completely conserved. There's a lot of variation in that family, even at this level that I sort of indicate here. But there's these 4 positions that are really important. Now when you look at these 4 positions in 3D, and those are 2 different sides of the same molecule, you see that the first here and the last come close on the surface. They're next to each other. The other 2 here happen to be on the other side of the molecule, also next to each other. Now, what that means is something remarkable. Imagine you cut out a window of 5 consecutive residues here, corresponding to that residue. Now, if you want to change something in this residue, it will have an effect on that residue. Because what survives is the 3-dimensional object. And the 3-dimensional object, say you made this one here a positive charge from a negative, then you may completely neutralize that by switching this one the other way around, if that were a salt bridge. You maintain the salt bridge, you maintain the overall charge, you don't change anything, you just switch. But that means that you actually influence something that in sequence space is here, much further away. And the probability of being able to see that, you see in this 5 residue window, in the profile of the 5 residue window. So this cutout 5 residue profile tells you what happens elsewhere, because what you evolutionarily observe in that window of 5 is what has happened to that object. You don't understand how it happened. But somehow this influenced the way it looks. You may not be able to get that information out, but it's there. In some sense, it's like you look at the raindrop and you see the world in the raindrop. You have a tiny cutout of the universe. And in this tiny cutout, you see something that happened elsewhere. It is like you go from a problem that is like a chaotic or a difficult problem here to a much simpler problem by averaging over different species, by averaging over different solutions that life found in some sense. So you make this complicated problem into a simple one. That's in principle the idea. The way to do that in the language of the neural network is simply, so again, I have the protein sequence running here. In my previous example, I showed this stretch here where the proline was in the center as the center of that neural network here. Now I replace the sequence here. I have an alignment. So this is protein 1, 2, 3, 4, 5. And at every single position, instead now, what the input here was 21 unit vector, where the 21st was the spacer, and the 20 were the amino acids. And that was a sparse vector, meaning that there were 19 zeros and 1 1. Now I replace this 19 0, 1, 1 by a simple real valued vector. And the real valued vector here could, for instance, have the frequency. At every single position, I put the frequency of the count of D in that particular position, and so forth, and so on. Clear? So I replace a single sequence, a vector with made up of sparse coding 1 and zeros, into a vector that is real valued, and simply puts the frequency of one of the amino acids, one of the 20 amino acids, in that particular position in the alignment. So in this example here, the D is 100%, this is 100%, but here we have the first change. There's an A and an E. And in fact, there's 3 A's, 1 E, here's 50-50, and so forth, and so on. So the rest of my system will be exactly the same. It makes the story a little bit more complicated. Now, if I want to predict secondary structure, what I have to do is I need to first query my sequence database. That's exactly the problem we talked about at the beginning of today. Finding with a blast, for instance, that in fact is the blast. I find a set of proteins, I align them with the dynamic programming, I apply this curve that tells me when are similarities such that I can infer similarity in structure, take everything above the line here, build a multiple sequence alignment, count the frequencies, and now put that into my neural network, where in this particular neural network I have again these two levels. The sequence to structure and then the structure to structure neural network. I can use some additional information that has to do with the overall length and some other aspects of statistics, and that is simply the same thing. Now, here's another thing. I said that the neural network essentially introduces hyperplane in space. And I'm going to show you now the hyperplane in 2 dimensions is a line. And what I see here for classification of the red and blue dots, that the gray ones here are particular solutions of neural networks. They are trained in a particular way. And you begin with a randomization of the initial values, you get to a solution, and say you get the gray solution here. And then there's another one. You start slightly differently, you have a different number of hidden units, you use some little bit different input information, you get that line. So in this particular case, I have 4 gray lines, and I can simply average over those, right? I can have 4 different, in this particular case, 4 different gray lines mean 4 different networks. And the way to average over those, how would I average? Would I average over the connections, or what would I do? So I have 4 different neural networks, and I want to somehow find a jury, a common thing. I want to average over them. How would I do that? A machine learning device. So the neural network, the free parameters are the connections. It could be linear regression. So take the free parameters. Should you average over the free parameters if you want to average over different solutions? Or what could you average else? Could you average over? Yes, the output. What is better? So you should clearly average over the output, the prediction in this particular case, because if you interfere with the model I don't really have, here's still the, sorry. So averaging over the connections means that essentially you have an average over this in 4 different networks, but in the 4 different networks it means something very different. And by taking out, going back into the parameter space, you mess up the system. So you absolutely have to stay in the level of the prediction. Now, is the green one going to be better than the gray ones? If you have 4 different machine learning devices, and you average over those. Here, I average over 4 different networks. And in fact, the reason why I have to average over 4 different networks ultimately comes back to this issue. Remember I told you that I have a balanced and an unbalanced version? One initially I said is 60, one is 62. Now, both have their strength. 62 is a higher number than 60. But the 60 lower number one is better for beta strands. So somehow, the best of both worlds, I just average. But now my question again, I have different machine learning devices. And since I don't know which one to take, I just take the average. Is that an okay procedure? Weighted average. Well, so what I dislike about the weighted average is that you introduce another parameter. But weighted or not weighted. So yes, if you want a weighted average with a weighted one. Okay, let's get back to. So we have two options here: weights of one, or you tell me how to choose the weights. How would you choose the weights?
Depending on the prediction of different.
So predicting. What do you mean by pretending on the prediction?
Like depending on the accuracy.
So she says, and that is a very, very good point. So you would, in fact, that's a very important point. You would, in fact, put a weight that is proportional to the performance of the particular system. So if you have one machine learning device that is much worse than another one, you can still use it as long as you downweight it. And there's a certain probability that then it will still, in some cases, contribute the right solution. And that is true. Let's get back to my first question. And let's assume that we have three, or in this particular case, 4 devices that essentially perform at a similar level. So weights, equal weights would be the solution there. My question to you, will the average over 4 always be better than each one of them? Or let's get back to the more provocative statement, then the best one. Okay, in this particular case, I say they're all of equal size, but one is a little bit better. Will the average be a little bit better than the little bit better one, if they're almost equal? And what's the condition for answering that question? Can you imagine what you have to know, what you have to ask, what you have to look at? Again, my problem always has 2 aspects. So I make mistakes. And there are always 2 sources of these mistakes. One source of the mistake is that there's something of the problem I do not address. Or something that is systematic, not in my data. No matter what I do, I cannot solve that problem. Then there's something that's a random mistake. White noise. This part of the white noise, by averaging over many, I will always improve on. So the degree to which I win by combining different devices has to do with how much white noise, how much of the mistake I make is white noise. The more it is, so the more the white noise is responsible for the mistakes I make, the more averaging really helps. The group of Søren in København has, a few years ago, really brought this to the extreme. They averaged over many thousand of networks just because they had too much CPU time, but it still helped. So you can really, as long as you have white noise, you can really drive this to the extreme. And typically applications, this is not the situation you will have. But what you will have is that you will develop different methods, and that at the end of your project you often will not quite know what to take. This is one thing to immediately try. Be careful with the weighing, because this is another free parameter. You have to be careful how you handle that. You have to make sure that you do the optimization of the weights on a cross-training set, not on the final set. Okay, now we get into what else can we do? So we have evolutionary information, we have 2 levels of networks, and there's something else we can do. Let's skip that. When we do all of that, the system gets to the performance that I show here. Again, that's the sequence shown up here from residue 1 to residue 57. That's the observed secondary structure by DSSP. So you have a 3-dimensional structure done by crystallography in this particular case for the SH3 domain. You translate that 3-dimensional structure using DSSP, hydrogen bond formation, into a string of secondary structure assignments, E, 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 a short 310 helix, and E. This is first generation Chao-Fasman. This is Gore, second generation, Gore 3 it should be. And that is this method that uses the profile as I show. For Chao-Fasman here, this is an overestimate, so this performs actually better than its average. This, for Gore 3, for the second generation method, also is an overestimate, 65, by at least 5 percentage points. This also by about 5 percentage points. This clearly performs better. So for this particular example that I show, in fact, I completely got over the magic mark of 65. Using profiles and using all of these tricks here, I got to a point that was had previously been believed to be unreachable. Now, is that one example for which it works? The answer is here. That's the distribution of many proteins. Performance, the 72 is pretty much in the middle. Okay? So that is in fact a method that predicts on average much better than anything before by just using profile information. And to repeat, profile allows you to bring more information into the same input window. You still have only 13 input units. Nothing has changed. You increase the information density by simply focusing on what is important in the family. And that you got from aligning sequences. So you get your information from somewhere else. But the machine learning device doesn't really change. Now here's another important aspect. And that again is something that is true for any type of machine learning device. Or any type of, I'm sorry, I'm missing a slide here. Why didn't I see that? So my neural network has the unit H, E, and other. I usually call it L here. And say my network has the value 0.6, 0.2, 0.2. And by the way, they don't have to sum to 1. That is one network, and this is the second network here. 0.9, 0.05, 0.05. Both of these predictions are helix. In both cases, the network would predict helix because that is the highest signal, right? But you immediately see that there's a difference between these two predictions. This one here is much stronger than that one. And that strength I could translate to some, let's call it reliability. I could see whether that reliability, which if you want is a prediction of how well you predict at every single point, because that value has nothing to do with knowing the answer. Just from your output you can deduce this value, the reliability, and that in fact is exactly what I show here. You can't see that as a light gray, but you may be able to see these green stars here, and the green stars are drawn whenever this reliability is higher than 50%. And for every one of these green, the prediction actually is right. So can I then use that to possibly guess in this distribution what you see? Yes, the average is 72, that's great. Sometimes you're even doing better, that's even better. But also, sometimes here, there are cases where you're way, way, way below random. So for this particular protein here, a random prediction would be better than my great prediction device. But the problem is you cannot know it ahead of time. And clearly this 72 is much better than 30. 35, much better than random. So you clearly don't want to use the random prediction for everything. And how can you guess that for this one here, random would even be better than my prediction method? And the answer is, hopefully, in this reliability index. What I show here is the performance per protein, and what I show here is the average reliability index of an entire protein. And you see that for some cases where the prediction is low, this tends to be low. And for proteins where the prediction is very high, this tends to be high. So the reliability index gives you some idea on which side of the distribution you are. Gives you some idea about how well the prediction really is. And here's another aspect of that. What I show is the percentage of residues predicted versus performance. The line that is important here is the dark one here. And 72, so if I predict for every single residue, then the performance is about 72. That's what I talked about. If I only looked at the residues that are predicted above a certain level of reliability, say only at roughly 50% that are 7 or higher here. That's roughly 50% of all of them here. Well, it's a little bit less, 40 something. 45% reaches a 7, and for those, the performance is 88. Again, for that to pick this 42% or 45% of the residues, I do not need the structures. I just need to look up how strong the signal is, the probability of my neural network there, the probability of making the decision. That's all that I show here. And I overlay it in this particular graph. I simply explain to users what that would imply for the expected accuracy. So I put two things together in the plot, but for the user, in fact, I can give the reliability index without ever knowing the structure for every single prediction. This is something that you can do with almost any machine learning device in almost any problem that you have. And it's completely underappreciated. This is one of the most important things that you can contribute to people, that you give them a feeling. We always do things that are right and wrong. Certainly when we do predictions, when we do machine learning, when we do data mining. Give the people a feeling what is most likely right, what is possibly wrong. They have to guess that. Give them, in the way you write the code, steer them toward thinking about that, considering that. And one very important problem that everybody has to ask is, I know some things are wrong, which ones? I know some things I can trust more, which ones are those? And again, as a programmer, as a developer, it's your task to help people to find that. Most people, again, don't do that. And this is exactly the instrument that you need in order to do that. Let's skip that. Now, here's an example. It is a little bit complicated slide. Let me tell you the background story. The background story is you begin with an 11-mer, a peptide, a stretch of 11 amino acids. That looks like this. In some protein, it's in an alpha helix here. I put all of that in green because that is the beginning of the experiment. And the experimentalists who did that, Kim, I'm sorry, I'm missing the, up here it is, Kim and Burke. In this paper, the experimentalist had the challenge to somehow change that stretch such that in one position of the protein, it's an alpha helix, and the same stretch of 11 amino acids, and in another position, it's a beta strand. And that, in fact, is the solution. Here, it's a helix, and here, the same 11 residues are beta strand. Okay? Context dependent. Now, what the slide also shows is the secondary structure prediction method. And the secondary structure prediction method, actually, the one that uses profiles here is the second one. The no one is the ones that does not use profiles. The no thing here predicts here a helix and here a helix, because essentially it only sees sequence. It has 13 consecutive residues. Once it is a helix, the other one is a helix too. So the same residues, same prediction. While the one that sees a profile sees from the environment a different signal here and a different signal here. Therefore, here it gets correctly the beta strand, and here correctly the alpha helix. That again is a dramatic example where, so the window size that has been used here to generate these predictions was 17, not 13. It's longer. Actually, it's a mix. But still, you see that the core of 11 residues is identical. Still, you get an environment signal, and you get that environment signal largely from the profile. That's remarkable. Okay, let's get back into this theme. And that is another theme that is clearly has been part of most examines so far, because I believe this is a very essential part, not so much of computational biology, not so much of protein structure prediction, but really of assessing methods. And computational biology just has a necessity to better learn to measure. In the case that we have method A being 60% and B being 63%. Can you say that method B is better? You have to, there are a bunch of questions that you have to ask before you can, in fact, answer that question. The first one is, when the author says 63 and 60, are they actually measuring the same thing? Is one maybe talking about the accuracy of predicting strands and one of predicting helices? Or is one looking at all residues? Is one, in fact, looking only at better predicted residues? It's very, very important. And the remarkable reality is that in possibly more papers than the people compare apples and oranges, so they don't compare the same thing. Okay. Now the next question, given that they in fact assume that both of them measure this Q3, so the number of residues, the percentage of residues correctly predicting either of those three states for both of them. What else do I have to ask? Well, the next thing that people again very often don't realize is they need the same data set. If the one method is evaluated on one set and the other one on the other set, then the numbers are not comparable. This happens because everybody develops a data set today, whenever they publish, and they typically take all the proteins they have. When I put up my method today, number 1, and in 2 years from now, you put up the competitor and you want to beat me, then in 2 years from now you have a new data set. Rightfully so, because in 2 years there's just so much more data and you ought to use it. But you cannot compare one-on-one without, in fact, translating my method to the same data at some point, or back tracing your method to mine, and ideally my method to your data set. Now, the next issue is, say both use the same data set. Most of the time today, predictions have to do with some parameter fitting, some machine learning. So the next question really is, how are they split? How is cross-validation done? Is it done the same way? Or is it true that in fact one, yes, we all had the same 100 proteins, but one used only 30 for training, only 30 for testing, and the other one had everyone used for training, for testing once. And in fact, again, we have to see an example where every method has used every one of the proteins in the set available for testing exactly once. Not more often than that. You cannot randomly sample. It is very, very important how the split between training and testing is done. And this is unique to biology. You have to be very careful, mindful about the redundancy between data sets. So the background here. We want to predict secondary structure because we cannot predict 3D structure. We cannot apply comparative modeling. We want a methodology, we want a method that predicts something that I cannot otherwise do. So my criterion for splitting between training and testing is very clear. If I can do comparative modeling between training and testing set, nobody will use my method because then we can predict 3D structure. So testing and training sets have to be so far apart that I cannot apply for any pair comparative modeling. That means they do not have a significant sequence similarity, and significant sequence similarity could be measured by that. So any pair that is in the blue region, pair of proteins cannot equally at the same simultaneously be part of the training and testing set. I will bring this topic up again in the next lecture, and I believe I will stop here. Are there any questions? Again, please, for these lectures, try to come up with questions that you ask. I will continue the system of going through a couple of questions the first 10, 15 minutes. Hopefully that will improve over time. But still, please come with questions. If you have questions about the IDP, come and see me. If not, have a good weekend. Thank you.